So as I was saying, please go back and check your exams, look through those osmosis problems, the one with the beakers and the ones with the pictures of the cells. And if you don't understand exactly, do you have kids, Dorian? And if you don't understand exactly why you got those wrong, come see me, okay? I can make it simple for you. And as I said, the essays were pretty well written for the most part. You know, some of you, okay, but anyway, come into class and y'all here are the ones when I think about some of the better essays I read. That really helps, obviously. Remember, the withdrawal deadline is not for another 2 weeks, so you do have some time. We have a lab practical in between. You still got, I think I added up today, something like around 600 and change points left. So 2/3 of the points are left. So you haven't done as well so far. It does not mean the end of the road for you. It means some folks it's mathematically impossible, or pretty close to mathematically impossible. But just because you're looking at your grade going, I don't, you know, you still got a lot of points left. Remember, there's a lot of those smaller assignments. Do well on those small assignments, folks. Take your time on them. Remember, I said if you do them and you want to try them, like if you bomb them or really do poorly on them, or you just misunderstood them, I'll give you a chance again. I'm not going to let you scrounge points. You know, you got an 18 out of 20. Can I try it again to get a 20? No. That's not the intent. But, man, if you go through something and it's just, ugh, but, well, of course, I guess I'm just setting up for that human behavior where you bomb it on purpose just so you can get the answers. But, you know, I look at that, again, as trust and understanding that on the exam you can't do that. And I use those assignments to help you get ready for exams. All right? So take a look at those. What have we got in the schedule? We don't have our lab practical next week, right? Right. We do not have it next week. We have it the week after. Do not have it next week. It's the 16th. We have it the week after. Correct. So we're doing lab. I think it's 8. I'll post it. It's whatever the next one is in the morning. Hopefully we'll get it. I'll get it the right way this time. We're in the middle of changing and updating the labs, and that's why sometimes the labs go a little bit screwy. We're kind of learning ourselves to use this new program and all the things it can do. No, we are. We're just not doing the lab practical. Sorry. Okay, I got to get started on this because there's a lot to talk about in this chapter. It's really one of my favorite chapters. It's funny. It's one of the chapters every once in a while we have to go through a process where a faculty member, a peer member, another faculty member, comes in and evaluates your course. So you do it for like any job, right? And they evaluate a lecture, and oftentimes we'll say, well, what are you guys talking about? And like, for example, my buddy Matt, Professor Wood, when he was talking about viruses, I'm like, cool, because I don't know crap about viruses. But when everybody says, well, what's your favorite lecture to give? This is always one of the ones I say. I love talking about this. And then in the bio 2, I love talking about tetrapod evolution as we crawl out of the water. Anyway, another story. If you look at this picture here, you might think, why would this guy find this exciting to talk about? Well, for one thing, it taps something in me and maybe in all of us about learning how things work. I am no engineer, trust me. I'm an average guy when it comes to taking things apart and putting them together, or, you know, and that kind of stuff. But I'm not designing bridges for you. But what we're going to do in Chapter 7 is essentially we're going to talk about what you see in that picture. And if you're wondering what the heck that is, this is basically a chemical reaction schematic, okay? And you've probably seen those before, especially if you're into electronics. You know, schematics are how things are hooked up, right? Like how wires are hooked up. And the little round circles that you see there might represent the juncture between two wires if we were looking at an electronic schematic, right? Well, this isn't an electronic schematic, like I said. It's a chemical one. What this is, excuse me, this is what we call, and you see the title, metabolic pathways. This is how organisms, not just you, right, but this is how organisms take the energy that's available in their environment, not the energy they make, because you can't do that. You can only convert. This is how they take the energy available in their environment. And energy could be you know, plants, eating plants, or eating things that eat plants, or, you know, all the things that we know about, and using that energy, not destroying it, not using it up, not getting rid of it, but converting that energy, taking the energy from the bagel, from the rabbit that you, I could never understand how anybody could eat rabbit, but when I lived in Greece, There was a market down the road. My mother hated it because there were rabbits laying out on the market there for sale, fur and everything. Just like popped them on the head and threw them in there. But anyway, the energy from wherever you're getting it, rabbit or otherwise, does my pen work today? Let me see. Yeah, oh, it does. The energy we're getting from whatever we're eating is the energy we're doing this with. Remember, in thermodynamics, last chapter or so, I emphasized to you, at least I tried to emphasize to you, that the fact that these reactions are coupled, that exergonic and endergonic, that catabolic and anabolic, and then even way back, reduced and oxidized, these things are coupled. You can't have one without the other. It's a cycle, right? So the energy you release out of a bagel, let's leave the rabbits alone, is exergonic, right? The energy release out of the bagel, that's an exergonic catabolic reaction. You're tearing the bagel down. You're tearing down, as you're going to find out today, you're tearing down the little molecules in the bagel. And you're using that energy to do something that requires energy. Now, you help me out here. Why does this reaction I'm writing, the way it's written, as you see right there, why do you know, what tells you that it's an endergonic, or most likely an endergonic reaction? If you understand what ADP is versus ATP, which you should, right? It's 2 phosphates or 3 phosphates. How do you know that's an endergonic reaction? How do you know that's an anabolic reaction? Yeah, you're making something bigger, right? Anytime that get off the chemistry, right? Just think about it in general. Anytime you make something bigger, anytime you synthesize, anytime you do any of that stuff, it's a law of thermodynamics. Whether you're a rock or a prokaryote, you need energy to do that. So you can't make it, right? Even plants don't make it, right? That's the sunlight energy they're converting. But what we do is we release the energy from food in a catabolic reaction, and we take that energy and we use it in a reaction that needs energy because it's building something, an anabolic, an endergonic, so exergonic drives endergonic. And then that ATP, where's that go? It goes to do this. Or write about me doing this, or to write about me talking about all this. Now, before you freak out and look at this and go, he's nuts, we're not going to go through this entire, what I call chemical schematic, right? But you can see, like it talks about how chemically your body would digest lipids. There they are down here on the left. So there's a variety. Your body is extremely variable. It's got a lot of different switches it can hit when it needs to do metabolism. We're going to focus our attention for 99% of this chapter on this stuff that's running down the middle. It's got a little loop at the end. We're gonna focus our attention on carbohydrate metabolism. And the reason why we focus on that when we're talking about respiration is, and I'm really simplifying it, but chemically speaking, that's right. Yeah, that's an endergonic reaction, endothelium, because it's not a P1, it's a PI. It means inorganic phosphate. But the important part is it's taking a thing with 2 phosphates and making it with 3, so you're building it up, right? So what I was saying about the reason why we focus on the carbohydrate is more or less, and if you kind of look at the schematic, you can see it. You're welcome. More or less everything else that you metabolize, or anything else that's involved in metabolic pathways, like things like, I like this one in the upper right, biodegradation of xenobiotics. I don't have any idea what that is. Xenobiotics xeno means foreign things. So this might be the way your body's immune system works. Could be. Anyway, the point is, it's sort of like carbohydrate metabolism is I-4, and lots of roads kind of lead onto it. So there's many different ways to get from Tampa to Daytona on I-4. You don't have to start in Tampa or start in Daytona. You can enter in Lakeland or Orlando, blah, blah, blah. You get the idea. So that's why we focus on carbohydrate metabolism. Plus, it's a heck of a lot easier, right, isn't it? So we're going to focus on the blue part in the middle. We'll talk about why there's a circle, too. It's really cool stuff. Talk about learning how something works, right? Now, I'd be remiss to one of my best college professors that I had in graduate and undergraduate, ecology professors, really neat dude. He was a big runner. Every once in a while, I see him out still running, and I'm like, my God, he's got to be 90, and it's almost 90 outside. But he's a really cool guy. But Let's talk about this idea of respiration and sort of a big picture, right? Big picture in the ecosystem. So what we're looking at here is an ecosystem. Now, I'm not going to tell you anything here you don't know. You know this happens. Plants convert sunlight energy at the top into food. Now, if you're looking for where it says food, that's what food is right there. Food. Thank you, pen. Always screws me at the wrong time. Food is our organic molecules, carbohydrates, organic molecules. Aren't they? Isn't that what food is? Proteins, carbs, lipids, nucleic acids, right? But they're all organic molecules. Now, the thing about organic molecules is, number one, they contain a lot of carbon. We love carbon. Can't be organic without being carbon. And many of them, and carbon is a great structural molecule to build things with. That's why we like to use iron and steel. It's got a lot of we use steel. Steel is iron reinforced with carbon, because carbon makes things kind of get rigid and flexible. Boy, I got off on metallurgy there for a second, didn't I? Organic molecules in living organisms represent the carbon, but energy. All right? There is energy, potential energy. Remember, potential energy can be chemical. A gallon of gas has energy in it. Potentially, you throw in a match and you turn that energy into another type of energy. What we call the kinetic energy, the fire, obviously. So in food, there is energy, right? Obviously, there's calories. That's why you eat it. No big deal. And so we take those those organic molecules, and if you look there in the equation, we oxidize them, don't we? Isn't that what that means? Where am I getting that from? Where am I getting the statement that what we're saying right here is that organic molecules are being oxidized in your body, specifically in your mitochondria, which we already talked about. But why is that oxidation? Or maybe why would you predict that's oxidation that's going on there? Organic molecules plus oxygen is doing something. Why would you predict that's oxidation? Yeah, it's breaking down, right? Which we've been talking about. And holy cow, as I said, not always is it oxygen is an oxidizer, but here you go, oxygen's being involved. So my first prediction would be, oh, I see what's happening. If we kind of follow down the arrow towards, say, I don't know, the 6 o'clock position, we see what's happening, right? ATP is being made. Now, how is ATP being made? Does anybody want to draw it on the screen for me? How does that ATP down there at the bottom get made? What is the chemical reaction that does that? I've wrote it a bunch of times already. Somebody do it for me. On the screen somewhere. Well, correct, and that's endergonic, isn't it? That's a building up of a molecule. So what we're going to need is the energy. The organic molecules have the carbon in it, as I said. Who wrote that, by the way? But we're also going to use the energy that's being released from when you oxidize things. You see something that we really didn't focus on. Thank you, Seth. Something we didn't really focus on, which we'll now. I'm going to come back to this slide, so don't worry. When we talked about oxidation and reduction, remember that back, chapter 2, I think? We talked about sodium losing an electron and chlorine gaining an electron. And we said losing electrons called oxidized because oxygen is so good at it. But it's the process that's oxidized, right? All right, so just take that same sort of concept. Let's go down below and look at this thing that looks a lot bigger, right? On the left-hand side is glucose, sugar, energy. We say glucose is the energy of choice in cells. Again, that's why the carbohydrate metabolism was kind of the middle of all that spaghetti stuff that we looked at the beginning, that schematic. Because glucose is kind of, that's what your body wants. Little nuggets of glucose. Your mitochondria eat up, degrade, catabolize little nuggets of glucose, which we'll get to. So that's glucose, that's C6H12O6. It was the ring thing we looked at in lab, the ring structure. All right, so take a now if you know about balancing equations, you understand why it's saying 6 oxygens and blah, blah, blah. I'm not going to ask you to balance equations, so don't worry about knowing how to do that, but that's what that is. But clearly what's going on here is oxygen, 6 oxygen molecules, are oxidizing glucose, right? Now, when something is oxidizing, something has to be reduced. So oxygen is oxidizing glucose and being reduced itself. So let's follow the bouncing electron. So the electron goes from glucose, okay, over to carbon dioxide. That's the electron. Now, here's where it becomes interesting. Later on down the road, and we're sort of skipping it a little bit here when we look at the way this whole works, this electron that we take off of the glucose, we're going to find out is not only just your everyday run-of-the-mill electron. It's actually a you know what? I didn't drop the electron. It actually goes all the way here. Excuse me. So the electron is actually just not every day a run-of-the-mill electron. It's a high-energy electron. Energy by virtue of the way it's built. Just like the electrons in gallon of gas. Gas is hydrocarbon. Carbon and hydrogen. It's an organic molecule. By virtue of the way that's structured, there's energy involved. By virtue of the way glucose is structured, those electrons are being removed, are not only being moved around, but they have energy. Now, they're going to be moved around, and they're going to be used to do something with them. They're actually going to be moved around, and it's the energy in them that's going to drive a reaction, right? So now we're kind of combining the oxidation and the reduction. So when the electrons come off of the glucose molecule, they go on to the oxygen molecule, right? So the electrons are moving over to the oxygen molecule. Now, for reasons that'll become apparent as we go later on, the hydrogens that those electrons were stripped off of, see, really what comes off of carbon dioxide or glucose is this. Follow it, do alphabet soup, right? C, H, O plus O gives you CO and HO. I'm just taking away the numbers, right? So very simple. If I asked a third grader, how do we turn CHO into CO? You say take away the H. How do you take O and turn it into HO? Put the H on it. So this is what's happening. Basically, this is exactly what's happening in respiration. Something with a CHO is being turned into CO and HO. Carbon dioxide in that metabolic water. This is a little bit of metabolic water that's given off. This is not the water you drink. It is chemically speaking, but it's not enough water to sustain you. It's water by virtue of the reaction. There are a few critters that live in the desert, you know, where there doesn't rain a lot, where this metabolic water provides most, if not all, of their needs. There's actually a species of fish that lives in a desert where it rains once every 10 years. Can you believe that? There's a fish that lives in, I don't know, I think it's somewhere in the Kalahari, some part of the Kalahari Desert in southern Africa, where it only rains once every, nope, it's a fish that lives there. And when it rains and the puddles that collect, it rains for a while, the puddles, there's eggs down in the sand. They hatch. These little fish swim, do the fish thing, reproduce, lay eggs. The puddles dry up in a little bit of time. 10 years later, the eggs that they laid, the water comes back. So I think it was 3 weeks the rains stay around. So they live 10 years, or they hang around 10 years as an egg to spend 3 weeks. Mother Nature never said you have to live a long time, just have to live long enough to reproduce. Yeah, no kidding, that's kind of cool, isn't it? All right, so that's what we're doing, is we're oxidizing glucose, right? We're removing the high-energy electrons, and along comes the hydrogens with them, and we're putting those onto oxygen. So we're forming this little bit of metabolic water. Now, this is an oxidation reduction process. This is a molecule being torn down. Take a look, it's simple. On the left-hand side, glucose is more complex than carbon dioxide and water. There's more energy in them. So you can't destroy the energy. So the last thing we add is we say this is an exergonic reaction. Breaking apart food, inhaling oxygen, mixing that with the bagel you ate. That whole process that creates carbon dioxide and a little bit of that water is also this creates energy. Now, sorry. Steve, come on, pen. There we go. Releases energy, not creates energy, releases energy. right? It's an exergonic reaction. And again, I go back to what Seth wrote on the previous screen, that energy is going to be used to drive ATP. Okay, makes sense? This is the respiration equation that we're going to go through. We're going to spend the rest of the chapter talking about how specifically that happens. But that right there is the nuts of it right there. Okay. Now in the ecosystem thing, that carbon dioxide and the water that's given off, now the water that's made in respirations is a little bit misleading here. That little bit of metabolic water that you make, you don't make enough to give to the plants like it seems to show here. Any of that little bit of metabolic water we make, most of it we probably absorb ourselves. It's not a lot. But there's water and carbon dioxide in the atmosphere. You get that. And a lot of that carbon dioxide comes from us exhaling, and a lot of that water comes from the fact that there just is a lot of water. All right, so we get carbon dioxide and water, and we go through a process we'll actually talk about in the next chapter 8, right? Photosynthesis, right? And those processes are going to put those molecules together. So now we're going to complete the cycle. Then we'll forget about photosynthesis and we'll go on. So that process that you see as the arrow breaks up toward the top, right here, is a process by which we're assembling a larger molecule from two smaller ones, right? We're assembling this larger molecule. What kind of a reaction is that? When you take small and make big, what kind of a reaction is that? And there's a couple of words you could use actually to describe it. Got to have energy. What's the other word that Emily could have used besides endergonic? What kind of a reaction is going to need endergonic? I don't know if it's a synonym, but it might as well be. There you go. Thank you, Tulsi. Right, where's that energy come from then? Where's that energy come from to drive that endergonic, which needs energy, or anabolic reaction? Where does it come from? I go outside and I look up and I see a big yellow ball. Yeah, of course. What do you mean I don't know? Where's the energy? Look, it's right there. Light. What do plants need? They need sunlight, don't they? Yes, it comes from the sun. Of course, it comes from the sun. All right. More on that when we get to the next chapter. Let's go down here and talk about what's going on in this process of releasing energy from food. This is very, very cool. Which ones? Well, there are a few plants that are white. There's some that grow on campus. They're called Indian pipe. And that's correct. They don't need sun, but they live in a very strange relationship with the roots of the plants. But so yeah, they're right. Absolutely. Okay, let's go back to this electron thing that I talked about, right? The movement of electrons. So focus now your attention down here. Let's make sure these are the same basic idea, right? That electron I said that's being moved around, the one that's attached to the hydrogen, okay? The electron that's being oxidized off of glucose by oxygen, I said was a high energy electron. And if you've taken physics or anything like that, or even just if you know, that in order to do work, in order to make something, okay, you're going to have energy. Now, I've said this statement at least 1,000 times by now. I'm just saying a little bit differently, right? If you're going to make something, you're going to need energy to do that. So this whole process by putting hydrogen, their electron, and oxygen together, stripping it off of glucose, that releases energy. Well, it's kind of cool. And the process of actually putting hydrogen and oxygen together releases energy. Okay, but it also makes something else. Now, hydrogen is the most abundant element in the universe. Gosh, it's like, what I've heard, 99.9% of the element in the universe is hydrogen, and the rest of it is all those other ones that hang on the chart. So we get bukus of hydrogen. On Earth, at least, we have a lot of oxygen. So you get this question sometimes, well, gosh, why do we have a water problem? Why don't we just take this hydrogen, which is common as dirt, and oxygen on Earth? It was what did our meters, was it 21%, something like that we read in the classroom? We ought to be able just to stick those two things together and make water, right? And solve everybody's water crisis. And of course, chemically, that is possible. And actually chemically that's done. Look, that's what your body is doing. As I said, it's taking the electrons off of the glucose, but those electrons are attached to the hydrogens. They're specifically attached to these guys. And so the H, of course, right, is ending up on the oxygen. So that's what we're doing. That's what I mean. We're making that metabolic water. So wouldn't it be cool? We could solve our problem. Both of these things are abundant. We could do that. And like I said, this is a chemical reaction that we actually have harnessed, but it's a chemical reaction with costs, okay? Here's the chemical reaction on both sides. The one on the left is uncontrolled. The same chemical reaction on the right is the way we do it on our cells. Let's take a look at the one on the left first. What this is telling you is the hydrogen, and again, they're balanced in an equation, half an oxygen. When you put hydrogen and oxygen together, you get water. Okay, good. You can do that in two ways. You can do that basically all at once. So this is like throwing a match into a gallon of gasoline. And you can release that energy very quickly or even all at once and get an explosive release of that energy. It's actually what used to propel the space shuttle up into space. Not the solid rocket boosters on the side, but the big fuel tank in the middle. That fuel tank was full of hydrogen and oxygen. It split right down the middle. So up here it had hydrogen, H2 obviously. Down here it had oxygen. And what it would do in the back is it would mix them. Semi-controlled, if you will. But it would mix them enough at a rate that they would get this reaction which caused thrust. And the solid rocket boosters did too. This is what brought out the shuttle, folks. The solid rocket booster on one side burned a hole in the side of the tank, and that hydrogen and oxygen mixed faster than they wanted to. You know, think about it, the analogy of the gallon of gas again in your car. A gallon of gas in your garage throwing in a match releases X amount of energy. A gallon of gas in your car driving you to school releases the same amount of energy. It's just how fast it's released, right? So this process of, let's just take hydrogen that's real abundant, oxygen, put them together, make water, you got the explosive energy release problem. We've harnessed it to some degree in the space shuttle, but we haven't been able to do it enough to, just make this abundant, make water out of this abundant substances. However, this is where cells are very cool, because if you look on the right-hand side, your cell is like the internal combustion engine. Yeah, in the chamber of your car, you don't dump all the gas at once. If you have fuel injectors like spray bottle things. It sprays a little bit of the fuel with the spark comes from the I guess they don't use spark plugs anymore. I don't know, whatever. They sprays a little bit of fuel that's ignited, a little bit at a time. So you're releasing the same amount of energy, but you're doing it in a released amount. One of the analogies I use with students is living on a 10-floor building, let's say, and you need to get to the bottom floor to go to work. There's two ways down, aren't there? Jump out the window, climb down the stairs. Let's say there's no elevator. Either way releases the same amount of energy. One way, all at once. The other way, every time your foot goes down the stair, your act of your foot hitting the stair releases a little bit of energy. That's something we can deal with, a little easier than jumping out of a 10-story building. So on the right-hand side, what we're going to talk about today is how that's done. Once those hydrogens are taken off of glucose, that's where these came from, these H's, are mixed with the O that we breathe, that we inhale, all right? So those hydrogens are ripped off of the glucose, and then we split them into their protons or hydrogen ions, these guys right here, and we'll talk about what we're going to do with those later on. Don't worry about that. But we're going to use these guys specifically. The energy, why do they have energy? By virtue of their chemical structure. Just like a gallon of gas has energy. A bagel has energy. And we're going to release that energy, blip, 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 like it's showing you with the little pinwheel things, blip, a little tiny bit at a time. We're going to use that to make ATPs. Dink, dink. We're going to get the same reaction. Look on the left-hand side. You see the dependent variable, the free energy. Basically, we're releasing the same amount of energy in a controlled fashion. Right? Makes sense? Isn't that cool? So you see what I mean about it's this idea of stripping the electrons off of the glucose, but also the hydrogen. But what you kind of do, if you look at the picture on the right, exactly. You see, you kind of keep them apart for a while, the hydrogens, the protons and electrons. Then you reunite them at the end. See, at the end, you make water. a little bit different than you saw on the left-hand side, but you take the protons, the electrons, and then the oxygen. See, these things are missing their electrons, so you add them back, the oxygen, you get H2O, right? Here you go, if you want to see the analogy of jumping out the window. So what's going on? The electrons from food are jumping down these little stairs. So we're going to set them through a set of chemical reactions. That's what's going to be the stairs, right? We're going to send those electrons through a set of chemical reactions, through a set of redox reactions. It's almost like bucket brigade, like when the fire, in the barn, and they would get the villagers out there. It's working the same way. These electrons are they're passing. These electrons are being passed. It's going to be proteins, by the way, that does it. And every time they're being passed, something's being oxidized, something's being reduced, and something's being oxidized, and energy is changing hands, and energy is being collected. It's all chemical. It's all this chemical dance, and then we make a little bit of that metabolic water. Now, I got to introduce you one more character, or at least I got to introduce you to a character before we start talking about the actual process. Do we have any questions from this point? You understand? Yeah, exactly. It's all regulated. Has to be. Has to be. If hydrogen and oxygen mixed in your body the way they do uncontrollably, you'd ignite, you'd explode. That is not why people catch on fire. The ones where you walk in and grandma's caught on fire. I think that's probably not true. All right, let's talk about those things that move electrons around. Now, yes, so I'm talking about reduction and oxidation. I'm talking about that process by which this happens. But these electrons need a helper. They need something called a carrier. And these carriers are aptly called electron carriers because that's what they do. They're the ones carrying these green things. They're the ones that are going to take the electrons off of food and the breakdown from food, and they're going to move it someplace else. And I'll talk to you later on about where they're going to go. Yes, they're eventually going to go underwater, but we still got to go down the stair steps. But I want you to remember that while these are physically electrons, they are high energy electrons. They have a lot of potential energy because we're going to do some work with them, okay? So let me introduce you to the electron carriers. There are two of them. One is called NAD+, and one is called FAD. Now, there's a chemical structural formula of NAD down below. I'm not concerned with you memorizing that, but you might memorize. You might notice a few things. NAD's name is nicotinamide adenine dinucleotide. That's a mouthful, right? Let's just call it NAD. This is the adenine. So this is why you may recognize some of this because right here, this is the adenine. This is a ribose sugar, isn't it? This is a nucleic acid. All right, but this particular nucleic acid, the way it's set up, the important part is up here. It's kind of in the shaded part. This nucleic acid is very good at carrying electrons. In other words, it's very good at being oxidized and reduced, okay? Now, again, I'm not concerned with you looking at the chemical structure. If you want to look at it, you can see where the reduction occurs. You see there's one hydrogen there, and there's two hydrogens there. Hydrogens and their electrons. Always think that those hydrogens and electrons are going together. All right, here's what I want you to know, and I put it in red. The other electron carrier, by the way, is FAD, flavin, like riboflavin, the vitamins and stuff. But we're going to call these things NAD and FAD because I'm just not going to tongue twist myself over that all day. All right, what's down in the red is what you need to know. You need to be able to recognize, because you're going to see these on charts as we go through them, when they're oxidized and when they're reduced. And it's very simple. If there's an H to it, NADH, FADH2, then it's been reduced. Otherwise, it's oxidized. So on the left is oxidized, on the right is reduced. Get it? Any questions about that? That's going to be important because we're going to play follow the bouncing electron. Now my pen works good. Sometimes if I take out the battery and put it back in, it works. All right, now let's take all this that we've been talking and jump into the cell. Here we are in the cell. Here we are. The cell. All of this is the cell. We are specifically, this is like when you're at the mall and you walk in, you are here. Out here, this is the cytoplasm of the cell. Cytoplasm of the cell. The cytosol cytoplasm, so that's the liquid part. So there's a cell membrane around all this. And this thing that's kind of drawn in the background looks like a bean. That's one of those organelles we talked about, the mitochondria. Remember, the mitochondria is one of those weird organelles that may actually at one time been something that was free living a few billion years ago. The reason why we know that, if you can even see in this picture, it's basically a membrane around a membrane. Remember I said that everything in a cell is surrounded by membranes, but this thing's membrane around membrane. It's got the membrane on the inside, the one that goes back and forth, and then it's got the membrane on the outside. So it's almost like something's membrane got enclosed by a membrane. Maybe that was a critter. All right, we're going to follow through each one of these steps. We're going to go through glycolysis and pyruvate oxidation and citric acid. We're going to go through these step by step, and we're going to follow, as I said, the bouncing electrons. You notice where it's mentioning electrons everywhere. Actually, there are electrons that come up here too. So throughout this entire process, the entire from left to right, the big thing is we're going to be talking about gathering electrons. But I need to start on the left-hand side, obviously, first, and I need to let you know that it doesn't always go this way. In other words, what I'm saying is there's a juncture in the chemical reaction right there where I drew that line. Glucose being turned into pyruvate, no problem. Glucose being turned into pyruvate is a process known as glycolysis. Literally, the word means glyco, which is sugar, and lysis, you've heard before, is to split. So what we're doing in here, there's pyruvate. Remember from that enzyme the other day, pyruvate dehydrogenase, right? Anyway, what we're doing in glycolysis is exactly what it says. We're splitting glucose. We're taking that sugar and we're splitting that molecule in half, and we're making pyruvate. Now, that's going bigger to little, so we're releasing energy. That's the energy down here. Okay? Now, I'm going to come back in just a second, but I want to follow the other side, the bouncing electrons. This process is also going to remove electrons. You're going to oxidize glucose. You have to release energy. Can't release something without oxidizing. When you oxidize something, it releases energy, and the energy is not lost, but it's transferred to where the electron went, to where it was reduced, remember? So the electron is a physical thing, but it's also an energy thing being transferred. Okay, so let's talk for just a second about the process by which ATP is made. First two ways that we'll talk about, we'll get to this one in just a second, known as substrate level. They're very, very simple. You've already talked about it. We've talked about enzymes. Last one's a little bit more complex. I had a deaf student in my class a few years ago, and they had a sign language interpreter in there for her. And if you know anything about sign language, you know quite a bit of sign languages. A sign means a phrase or something. It's not just spelling. But there was no phrase for oxidative phosphorylation, that last word that you see in purple. So whenever I would say that in class, I felt so bad for that woman. Her fingers were going 100 miles an hour trying to spell it out. So I would try to help her out by just writing it on the board as well. I'm thinking, how do you sign oxidative phosphorylation? All right, so we'll get to that in just a second, but let's and that's how those ATPs are made, to oxidate. Phosphorylation, by the way, the word phosphorylation means to stick phosphates onto something. If I phosphorylize you, if I could, which I can't, I would stick a phosphate onto you. Now, if you're thinking, oh, I know something that's been phosphorylated, you're right. ADP is being phosphorylated. That's how that ATP there is formed. We're sticking a phosphate on something that has two, and giving it have three. It's simple math. But let's start with substrate level first. Out here in glycolysis, how are those ATP made? Just like this. Enzyme substrate. They bind together. Enzyme product. Nothing new there, right? In this particular case, the substrates are ADP and some other green thing that has a phosphate that it's willing to give to make ATP, right? And so the enzyme binds, and then they fall out, and ATP goes out and does whatever it needs to do, like wave its hands. I can. Which slide do you want me to go back to? That one? Okay. You'll see this again. These words and this flow, yeah, these words and that flow will come up again. Don't worry about that. I'm not going fast because I'm trying to confuse you, you know. Things that you see again, I just introduce you to, and we'll come back. So there it is right there again, right? Same sort of idea, but what we've done is just highlighted where we are. So this is literally like that thing at the mall. You are here. Again, there is a little, just a tiny little mistake. There are some electrons that occur right here. Hold on 1 second, folks. Dogs are getting. Cooper rummaging around in the dirty hamper. Oh my goodness. Then though those smells, like my wife says, why does he just love those? Like go outside and roll. And I said, you say smell, he says fragrance. So anyway, let's talk about glycolysis here. All right, let's talk about what happens. Let's talk about this sugar splitting, which is literally what the word means. Here it is. There's glycolysis. 2 sets of reactions, top and bottom. Okay. Simple enough to put into your head, right? Notice when you're looking at these words that some of them are words with endings that are familiar. ASE, those are all enzymes, aren't they? Now, do I want you to go home and memorize all of this step by step? My God, no. No, God, no, not at all. I don't care if you know that 3-phosphoglycerate by the enzyme phosphoglyceromutase is turned into 2-phosphoglycerate. I'm not concerned about that, right? This ain't a chemistry class. There are some things I want you to know what's going on. All right, let's start with where we are. Let's start with the start. Glucose. So you should be able to tell me where that comes from, right? That's food. And remember, it's being split down here in the lower right to pyruvate. These are all the steps in between. All right, so let's move through the first 5 steps. This does that, this does that. Oop, right down here, take a look. That's where it's split. Now, what this means basically here is we split the sugar into 2. One is the nugget we want to keep going, that the G3P. See that right there? If you go down below, that's where the reaction continues. The other one, that DHAP, that one is not, so we change it to the G3P. So we get 2 halves, one slightly different, but we convert that into the half that we want. All right, but so what I want you to know about not the name so much is the fact that sugar is split, but also take a look at these first set of equations, first 5. You'll notice that we're not making ATP here. We're not phosphorylizing ATP, are we? We're actually dephosphorylizing. So what type of reaction do you see here and here? Use your words. What type of reaction is going on there that's doing glucose into glucose 6-phosphate and so on and so forth? It's a reaction that requires energy. What's the word that we use to refer to those? Yeah, that's a catabolic reaction. Oh, you tripped me up there for a second, Victoria. I was thinking one thing and trying to say another, but you're absolutely right. That's a catabolic reaction. In other words, what we're doing is we're using energy. Notice I even put it up in red up there. This is the investment phase, so it seems totally counterintuitive to what I said. The whole idea here is to make ATP, not use it, right? Unless you remember back in our enzyme chapter when I talked about this thing, that little hill. Do you remember what that hill was called? What did we call that hill? We had a name for it. 2 letters. Sounds like. There you go. Got to prime the pump, don't you? That's what that is. So when we do glycolysis, to start with, we have to put in 2 ATPs. Keep that in mind, 2. Let's put that down there. Let's keep track of that. Because guess what? If this process ends up only making 2 ATPs, then it's kind of dumb. All right, so let's jump down to the bottom to G3P. Now, here's the first time you see our electron carrier friend. So tell me, what is the word that we use to refer to what's going on there? What's happening there? Now, if you're thinking maybe there's more than one word, well, maybe. Let's see. Dorian, how about you? Now remember, you got to know which one is oxidized, which one's reduced, right? So this one's oxidized. Remember, it doesn't have the H. This one's reduced. Reduced means you have electrons, so one does and one doesn't. So what must have happened there? Anybody else? I'm wondering if maybe I'm having a little bit of connection problems too. What? Yeah, NAD got reduced, didn't it, Seth? That's right, because it took electrons off of that stuff right before, whatever this molecule, the G3P, right? It took electrons off of it. Now, what's going to happen to those electrons? Well, we'll find out later, but I will tell you one thing about it. They do have to go somewhere. Oxidation and reduction have to occur simultaneously. They have to. Remember, I already talked about that in chapter 2. If you got reduced, if you got an electron, you got it from some other atom that got oxidized. You didn't go on down to the electron store and buy those electrons, okay? So we'll come back to what those electrons and where they need to go later on. But now, look as we go through, and notice the number by ATP. And remember, that's because we've split the molecule. So this is happening for each half. So each half is giving us an NADH. Each half is giving us an ATP at the beginning. Each half is giving us an ATP at the end. But add those two up. Add those ATP up. How many total ATP from the beginning do we make in the process of glycolysis? How many total do we make? Yeah, but that's the net, right, Seth? How many total do we make, though? Remember, we got to make sure. Yeah, we do, Dorian. We make 4. We make 2 for the first step, 1 for each piece. 2 on the second step, 1 for each piece. So there's 4 there. We use 2. So there is a difference. You got to remember the energy of activation. There is a little difference when we talk about how much energy is created and how much is yielded. Seth told me how much was yielded. See, that second part is called the payoff phase, and it's greater than the investment phase. So what you yield is 2, what you invest is 2, what you totally made was 4. Don't get so hung up in the numbers. Just keep in mind, it makes sense. You're going to need to get more ATPs out than you put in, or else it's silly to even do that. Mother Nature would never favor that. You don't get a lot of ATPs, and there are several organisms out there, many different species of critters. Now, I'm going to come off this page. Don't freak out because, again, you don't need to know the names of all these things, and they're in your notes anyway. But there's several critters out there that's where they stop. They only do glycolysis. In other words, these critters actually don't need oxygen. As a matter of fact, in some cases, we call them anaerobic. They're anaerobic respirers. Sorry, Jordan. But they're organisms that survive and respire without oxygen. As a matter of fact, some of them don't like oxygen because oxygen is a bad stripper of electrons, and so they die even in the presence of oxygen. Where are you going to find these? Like down in the mud, below a lake. Bottom of the ocean can become, the word is anoxic. And so these organisms can survive down there. You can also encourage organisms to do this kind of anaerobic respiration at certain times. So what I'm talking about here is the proverbial fork in the road, right? There's pyruvate. So in that "where are you" map, okay, we've moved out of the well, not out of. I'm sorry. We're still in the cytosol. But we got 2 places we can go. We'll talk in just a second about the arrow to the right, but I'm talking about what happens if you're anaerobic. That is, no oxygen is around. It's a process we call anaerobic respiration, and one of those is a process known as fermentation. And this is the products that they end up making. Let me show you how this works, though. All right, so we're staying in the cytoplasm. Now keep in mind at this first step of glycolysis, everybody does this, whether you're aerobic or anaerobic, whether you're anaerobic for a while, everybody does this. You and I do this. The difference is we go to the right. which we'll talk about in a second, but we tend to go to the left, which I'll get to in just a moment. So let's take a look at what happens if we take the path where there's no oxygen. So this is the same idea of glucose being turned into pyruvate, just a little different diagram. I know sometimes when you switch diagrams, it becomes a little confusing, so I try to set you up where we are. You'll notice that there's our ATPs. Obviously, we're talking about net. So we're taking that 2 rows of equations and just turning it into, well, basically what I want you to know, to be honest with you. And you should know there's enzymes involved in all this too, but I'm sure you know that. So you get the pyruvate. Notice down here, there's the oxidation part that we looked at. Okay, now, back to that for a second. When you oxidize something, you must reduce something. When you reduce an electron carrier like NAD, it's a carrier. It's just like we call airlines carriers. If you flew to Philly and you went on whatever airline, they'd call that an airline carrier. You can't stay on the plane when you get there. You're supposed to get off. You're not resident of the plane when you walk on. That's the vector moving you from point A to point B. NAD is the same thing with its electrons. They don't want the electrons. It just wants to move them. So as soon as it gets reduced, as soon as it takes the electrons off of glucose or wherever it takes them off, as you'll see, it immediately starts finding some place to put them. And that involves things called redox potentials and things that really don't concern you. But know this, that NADH that you see in the picture needs to put those electrons somewhere. Now, in the meantime, if we're talking about the process on the left, which happens in yeast cells, yeast are a fungus, and they're our friends, right? In the meantime, what happens in yeast cells is the pyruvate is breaking down a little bit. Some carbon dioxide is being released. Now, this is not the CO2 that you release, because you're not a yeast cell. When that CO2 breaks down, though, something magical kind of happens. Well, not magical, it's chemical, but something fortuitous happens. The breaking down of the pyruvate creates something called acetaldehyde, who loves to take those electrons from NADH, who now has some place to put them. So what does NADH do? NADH turns around and reduces acetaldehyde, and if you follow the arrow to the left, when you do that, when you dump those electrons off that you stripped off during glycolysis, you make something called ethanol. We call this process alcohol fermentation because that's how you make it. That's how you make beer. You take yeast and you force them to anaerobically respire. If you've ever been one of those home brew folks, you know that tub that you put all that stuff in to make it ferment, you've got to keep the lid on tight. And you also know there's a tube that comes out of it that you got to put into a bucket of water because gas is produced, and that's the CO2 that's produced right there. The tube's got to go in the bucket of water because you don't want air going back up through the tube. See, the yeast will switch over to aerobic if they encounter air. Well, when they aerobically respire, they don't make ethanol. You don't make beer. He's that buddy of mine lived downtown that would brew beer all the time. All right, now, if you're thinking to yourself, well, gosh, all I got to do then is hold my breath and I can get drunk, I'm sorry to tell you that you're not a yeast cell. But you do something similar to this, although it doesn't result in something that's actually good for you. It's wonderful. It demonstrates to me how life is this delicate balance. This process that I'm going to show you about has benefits in one regard, and yet it has detriments in the other. So let's get into it first. So this is how animals do the same anaerobic process, right? We get our 2 ATPs, we stripped off our electrons, we made pyruvate. Now, we don't have the intermediate step. We don't need pyruvate to break down into acetaldehyde. That's not where we exhale our carbon dioxide. There's another part of the equation you'll see later where it does that. As pyruvate is made, it's ready in an animal cell to be reduced, and yet then it does. So NADH swoops in and reduces it. Now, when you reduce pyruvate, you don't make ethanol, so start breathing, but you make lactate. Or another word for lactate is lactic acid. All right, now, what happens if in a muscle cell it becomes too acidic? What happens in any cell? What happens any place in your body if its normal pH is upset, if it becomes too acidic? What potentially, and if it's severe enough, will definitely happen? You do know. Yeah, you denature them, don't you? So here's what I'm talking about, the trade-off. Why would we ever do this? All right, here's when you do this. You're walking in the woods one day, and a female bear comes down the path, really ticked off at you. You got a little bit too close, and you better run, or what do they say? Lay still. Regardless, some wild animal's chasing you, so you're going to run. Now, ATP is kind of like milk in the way that it does not last really long. ATP is really volatile. It's ready to break down. You want it that way. You want it just on the edge of getting ready to give up its energy. You don't want to have to coax it out of it. Well, that means that you can only store a couple of minutes of ATP in your cells. Because after that, it makes no sense to make them because they just break down. So you shut down the pathway, which I'll show you a little bit later on how that's done. So when you start running from whatever the heck is chasing you because you're trying to survive, okay, if you've got to run longer than 2 minutes, you might think, well, then I'm out of ATP. I'm screwed. Well, no, what your body starts doing is taking food, immediately starts ramping up the process of making ATP. You got stored food. You got glycogen, which is in your liver. You got fats if you need it. Glycogen is much faster, obviously. You got glucose in your blood already. So you got stored food. So you're cranking that out. Now, there is a dilemma then. You got stored food, but you're breathing so heavy that you're not bringing enough oxygen to all the cells in, let's say, your muscles, specifically, or any cells, really, to let them be aerobic. Now, you're still breathing a lot, and you do have aerobic respiration, but some of the cells are now having to switch over to anaerobic. Why? Because there's no oxygen around. It's not available. You're using it very fast. Remember, you're running. Remember, all you got to do is outrun your buddy, though. Did I tell you about the dive knife? When my son, why do you have a dive knife? I said, stab you when I see sharks. All right, so you're running, and your body's cranking out food. In some cases, in some cells, it's gone over to anaerobic. Now, anaerobic respiration is doing exactly what it needs to do. It's making ATP for you. Man, do you need it, right? You're running. You can't make them muscles keep going if you don't have ATP. Now, is this the best way to do it? Well, you're going to find out, no, it's not very efficient to stop just with glycolysis, but you ain't worried about efficiency right now. You're worried about getting the hell out of Dodge. Now, there's a problem that's creeping up on you, though. The longer you continue to do this, you have to try to get away from that critter, and this is going to depend on the concentration of oxygen in the atmosphere and what shape you're in and all that kind of stuff. That lactic acid, that lactate is building up in your muscle cells, building up. pH is going down, pH is going down. If you don't get away from whatever's chasing you fast enough, you're going to start feeling muscle spasms, and then the next thing is you're going to be dinner. So let's say it's good news, though, and you get away. You're on the other side of the creek and whatever, you're away from the critter, and you're going, right? You're breathing really heavy. What you're doing, we say, the word, is you're paying back oxygen debt. There was a time there in which you expected your cells to respire, not expected, but chemically, they respired anaerobically because you did not have enough oxygen for all of them. So at the end, you're upping your oxygen intake to help get rid of that excess lactate. What that oxygen is doing is that oxygen is helping metabolize that lactate to bring that pH back to where it's going to be. Die or deal with this? It's an easy choice, right? See what I mean? Life is always that way. No. Run. Darwin, man, you always survive to the very end. Now, a little terminology here so we make sure we don't confuse. Never give up. Anaerobic respiration is when you don't use oxygen, is what we call the final electron acceptor. Now, I'll talk about this phrase again in a second, but what I mean is electrons have to go somewhere. We've already seen that in this picture. But if those electrons go into anything other than oxygen, that's true, if those electrons go onto anything other than oxygen, it's called anaerobic. Aerobic means, by definition, oxygen. That's why you do aerobics, because you become better with using your oxygen. If those electrons go onto something that's organic, like pyruvate or acetaldehyde, we say it's fermentation, but it's still anaerobic. If those electrons end up on something that's not organic at all, like sulfates, nitrates, sulfur, right? Things that aren't organic, things that live down around those geothermal vents that you see in the picture. This is a black smoker, so this is very hot, maybe maybe 500 degrees, very hot. There are organisms that live around there. And they're doing a process very similar to photosynthesis, which we'll talk about, where they're making food. And other members of that live down there are anaerobically respiring. They're not using oxygen, pyruvate, or acetylaldehyde, any of that stuff. They're using hydrogen sulfide, that nasty egg smell that you sometimes smell, nitrates, even elemental sulfur. And if you take my Bio 2 class, you'll learn there are even some critters who live off of digesting the iron. Oh, that's the only place they're found in the world, by the way. Digesting the iron on the Titanic. That's how they survive. They're actually eating the iron plates of the Titanic, and they're found no place else in the world. Isn't that cool? Yeah, it is. All right, so here we are, right? We stayed in the cytoplasm for just a second. We talked about anaerobic respiration. We talked about fermentation. We talked about what happens if you don't have O2. Okay, and as I said, we do this. This is the first step. It's what happens next that's important. That's where the fork in the road. So now we're going to take that fork to the right. Now we're going to go into the mitochondria. So now cells that don't have mitochondria, many prokaryotic cells don't, those cells cannot do aerobic respiration. So like in Bio 2, I talk a lot about some, excuse me, some protozoans who lack mitochondria, and they are anaerobic respirers. If you don't have a mitochondria, you can't do this last part, which has the name oxidative in it because it involves oxygen. As a matter of fact, here it is right here too. It doesn't involve oxygen this time. It involves a stripping of electrons, though. So let's go through that. Let's jump into the mitochondria. Now, let's step back for just a second before we jump into it. Sorry, I forgot I'd put this slide in here just recently from your book to kind of give you a bigger picture. This is the mitochondria. The cristae are the wound-up membrane inside. Like when I was skiing as a kid in Germany, like when you ski down the mountain going back, it was called stem cristae. That's the kind of turn you were doing. So this is where the membrane within a membrane, I mean, maybe it was a critter, individual critter at one time. But anyway, the result of all that packaging is we have these spaces. We have an outer membrane and the inner membrane. But what I really want to show you is the inner membrane space. So that's the space between the two membranes. And then the space way inside called the matrix. That's the blue part, okay? So when I use those terms in a little bigger picture, you'll know what I'm talking about. All right, so let's go back to pyruvate oxidation again. So here's where we are. There's pyruvate. There's the membrane of the mitochondria. So we're moving into the, don't worry about the inner membrane yet. We're just moving through that first membrane of the mitochondria. So what we're doing in this process, you'll notice, is carbon dioxide is being given off. That's some of the stuff you exhale, not all of it, but some of it. A coenzyme A is moving and attaching to something, but important for us, though, is what's going on here, right? There's oxidation. High energy electrons are being gathered. We even call it that. We call it high energy electron harvesting, like a farmer does, right? Harvests his crops. Now remember, back in glycolysis, since we're oxidation now, those electrons that would have went on to acetaldehyde or pyruvate, they don't go there. They're going to go the same place these electrons go. So we're gathering electrons. Now we haven't talked about what I do with them yet, but we will. But remember, these aren't just run-of-the-mill electrons. They're high energy electrons, right? Anyway, we're back at acetyl-CoA. Acetyl-CoA's got a sulfur in it. You can see all of this reaction that you see here has resulted in this molecule known as acetyl-CoA. Now, we're ready to move into the next step. All right. Again, there are some electrons that come off of here too, so that should be corrected. We're right here. This is where acetyl-CoA enters into something called the citric acid cycle. The citric acid cycle is called that because it makes one of the things is citric acid, this stuff in oranges, right? Same kind of stuff. And there it is right there, citrate. Now, again, this is one of those things where you look at it and you go, what am I supposed to do with all these names? You're not supposed to memorize them. But what you are supposed to know about this, well, I would like you to know acetyl-CoA. That's where we start anyway. But first thing I want you to notice about this is all these names that are going around are in a cycle, aren't they? So citrate at the very beginning turns into isocitrate, et cetera, et cetera, et cetera. And then at the very end, malate in the eighth step becomes oxaloacetate. Now, oxaloacetate is what you need to keep going. Isn't that clever? chemical reactions are almost like perpetual motion machines. So as long as you have acetyl-CoA and all these other things that we're going to talk about are happening, this cycle of reactions is always going to generate at the end what it needs to keep going. Now, this is not unique. Well, this is unique, but this happens more than once in Mother Nature. All right, down here at the very bottom, we got ATP. Are we back, folks? No, I saw my end where it looked like we lost connection for a second. Are we back now? Okay. Yeah, I saw my end where something flickered. I don't know what that was. All right, so what I was saying is let's start with the ATP that I said was made. You're going to make 2 more ATPs down here. 2 because, again, we've split the sugar in half. So this is going to happen twice for each glucose. And these ATPs are made phosphorylizing, but there's a little intermediate step, that, just so you don't get confused. GTP is guanine triphosphate instead of adenine. It's still one of those nitrogenous bases, just a little relative. But for whatever reason that I'm sure some biochemist could explain to you, there's an intermediate reaction. But you get ATP, just a couple, 2. Twice as many as you got with glycolysis, but we're getting ready to get a whole bunch more than that, let me tell you. Okay, so number 1, it's a cycle of reactions. You understand that. Number 2, you make some ATPs. But look, this is the real meat of all this. Look what's happening here. Look, some carbon dioxide is being given off too, right? That's what you exhale. What's happening in all those that I've circled? Oh, there's FAD for the first time. True. Right down there, right? FAD. So what's happening right there? What did I just circle? What's going on? What's going on there in those circles? I don't have my glasses on, so if you type it, make sure you type it. Oh, God, I can't see. Come on, what's going on in those circles? Not the same people each time. What's happening there? Tell me, what does that indicate when something goes in oxidized and something got reduced? That's what NADH is. They're all reduced. What happened there? Or am I losing connection? Ground control to Major Tom. Hello? Now you all got me worried. I got connection. Can anybody hear me? Okay. What's happening in the circles here? What reaction is going on here that I circled? Those are the electron carriers. What are they doing there? Don't worry about the names of the molecules, but what are they doing there? Okay. Not being rude is appreciated. Thank you. Now, can you tell me what's going on, though, with those circles, though? What is happening there? Yeah, it's oxidation is happening. Haven't quite got to the reduction part. Only something, something was reduced, but remember what's happening here, what Dorian points out, is we're gathering electrons. We're oxidizing things. We're harvesting electrons. What kind of electrons? What type of electrons, folks? What do we know about the energy level of these electrons? Why are they special that way? Come on, we're almost there, folks. Almost to the easy part. What do we know about those electrons? They have a lot of energy. Nice, thank you. So we're gathering a bunch of high-energy electrons. We did it back in glycolysis. We did it in pyruvate oxidation. Remember, there should be one there. We just did it a bunch of times in the citric acid cycle. And now the climax of our story is we're going to take all those high-energy electrons that can do work. We're going to take all that fuel and we're going to dump it into a process we call oxidative phosphorylation. It's just a fancy way of saying that we're going to stick a phosphate onto ADP. That's what that says. See down below? But the nice thing, or at least the really good thing about all this, we're going to make a lot of ATPs. All right, so let's take a look at how that happens. What am I showing you here is a series of particles found on the membrane, that inner membrane, that twisty membrane. That's why that membrane's so twisty. These particles that are going to do this work for us, that I'm going to show you how it works in just a second, are all over that inner membrane. There's tons, thousands, or hundreds of them making tons of ATP. But again, don't get caught up in the letters, the CYT, I think that means cytochrome, I believe. Don't worry about any of that. Again, things we need to focus on. Look right there. There's your electron carriers. Now, there's where they're delivering the electrons. There they go. See the stuff in the yellow? Those are the electrons. So now NADH is dumping off or reducing, in this case, FMN. And FADH does it just a little differently. And the NAD plus and the FAD are going to go back and gather more from the food you're eating. So now they're gone. Let's talk about electrons. Those electrons have a lot of energy. Look on the right-hand side. Free energy relative to O2 kcals per mole. Whatever. Up at the top, near 40, 50, those electrons have a lot of energy. So here's what we're going to do. We're going to pass those electrons down stair steps, just like we talked about at the beginning of class. We're going to go from FMN to FES to Q and blah, blah, blah. Along the way, we're going to encounter a couple of really big proteins called 1, 3, and 4. Now, those proteins are absolutely, well, all the proteins are important, but those proteins are important for a particular reason. But we'll get to those in a second. Let's follow the electron. As the electron is going through all of these proteins, these multi-protein complexes, it's transferring its energy. See, it's going down the hill, so it's releasing a little bit of energy at a time. All right, and it's going to do something there. If you're thinking making ATP, you're not quite there yet, but you're very close. We're not making ATP from this. Anyway, at the very end, here comes oxygen. Oxygen has kind of a anticlimactic role, really. All oxygen is, remember we need an electron acceptor, oxygen takes those electrons once their energy level is dropped down to whatever 20 means, 20 free energies. Once the energy level of that electron has dropped down to a point where it's no longer sufficient to do what it needs to do here, then oxygen takes it, hydrogen then combines with it, and we're back to making water. That's metabolic water. Now, that's why you need oxygen. You need someplace for those electrons to go. If you're doing this anaerobically, if you remove the oxygen, the whole system backs up. The electrons have no place to go. So where do they go? They go onto pyruvate, or if you're a yeast cell, they go onto acetaldehyde. You see how that works? You got to have some place for all those electrons to go. Now, folks, I'm going to tell you, I might run a couple of minutes late, like say 10:52 or 3. If you can stick with me, please do. I will keep the recording on, but I want to finish the chapter because we're in flow right now, okay? So if you can stick with me, that's fine. If not, I understand. It will be recorded. So anyway, I'm taking this picture you see right there, okay, and I'm transferring it to something that looks a little more like what it looks like in real life. So here's that inner membrane of the mitochondria. Here's that inner membrane space, not outside the mitochondria, but not in the, well, this right here is the matrix. Not in the matrix, the part that was blue in that other picture. I've mentioned to you a couple of times this semester how important membranes are. And I said that one of the reasons membranes are so important, like when we talked about diffusion and active transport, is you got to have something physical to separate things. See, Mother Nature wants to balance things out. If you could create a disequilibrium, you being a living critter, you might be able to take advantage of Mother Nature wanting to balance it out. All right, so that's what we're doing here. As those electrons, they're in the yellow arrows, are being passed off of NAD and FADH2. As those electrons are passing from these particles, remember they're releasing energy. These particles 1, 3, and 4, those are the special proteins. Well, they're all special again, but the important proteins are proton or hydrogen ion pumps. Okay? Pumps. They pump protons. They pump protons actively. They pump protons against the gradient. So what do they need to be able to do that? What does proton 1, 3, and 4 need to be able to move those red arrows of hydrogen ions from one side to the other against the gradient, so that sometime we'll get a situation where we get a boatload of hydrogen ions out here and just a little tiny bit in here. What do we need to do that? What do those 3 pumps need? They need energy, not ATP, Emily. We haven't made that yet. Remember, ATP is a form of energy. We do eventually make ATP, but this time the energy doesn't come from that. It comes from the electrons. Remember, the electrons are high energy, like from food. There you go. So the high energy electrons turn on the pumps, and they create a disequilibrium. Again, don't worry about it. It's like a bucket brigade. We're continually oxidizing and reducing down stair steps. We're releasing stair steps. We're releasing energy a little bit at a time. Remember, we don't want to blow up. All right? Now, at the very end, here's what happens, right? Oxygen, the electron, hydrogen stick together, make the metabolic water. Any questions on that? So that's the end of the trail for the electron. The electron's energy level is dropped. It has none left, but oxygen loves electrons. They'll take any old worn out electrons. So they go off into the sunset and become metabolic water. Any questions on that? All right, now the cool part, those protons. When we were pumping those protons across, we eventually got to a point, like I said, where there's a lot out there in the dark orange, and there's very few protons down there where it's yellow. There you go. So there's a disequilibrium, which Mother Nature does not like. She wants things in equilibrium, doesn't she? Diffusion is natural, occurs natural, right? Osmosis is natural. All you need is an aquaporin. But, you know, it's a natural tendency. So here's a situation where, like, you're taking advantage of Mother Nature by, say, for example, rolling the rock uphill. You're actually getting up to the top of the hill to let Mother Nature do what it would normally do, let it roll down the hill. Okay, fine. Maybe you tie a tractor or a wagon to the back of the rock and have it do some work for you. Let's go back to the protons and the pumping. Boom, boom, boom. We're pumping protons across. Mother Nature says, whoa, 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 we have disequilibrium. Now, to be fair, some of those protons, very few of them, nothing's 100% efficient. Some of those protons can leak back through the phospholipid bilayer. That's why when you see a number here in a second that says about, you understand that this is 100% efficient. But most of those protons go through a particle at the very end that I'm hoping you know is an enzyme, ATP synthase. All right, stick with me, folks. I am literally just about to finish. So notice what's happening in ATP synthase. The process called chemiosmosis, which is nothing to do with water, but it does have to do with stuff flowing from higher to lower. But the important thing is this is where the process of phosphorylation is occurring. See it? ADP to ATP. So this is something so cool. Let me show you. The next picture is ATP up close. Here it is what it looks like with a little more detail. It's actually a spinning rotor and a stator, and physically, this is so cool. When protons flow through that little opening onto the rotor, they cause it to spin around. Then they go out that second little opening there, and that spinning of the rotor causes this knob to turn, which causes this catalytic knob to turn. And every time it does, it's like one of those turnstile things at a concert. Every time that catalytic knob turns, another active site opens up for ADP and a phosphate, and then ATP is kicked out. So it's like boom, 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 boom. Every time that catalytic knob spins, every time a proton goes through and it spins this little thing around. Isn't that cool? It's a little tiny gizmo. Man, if you're into nanotechnology, that's awesome. All right. Now, numbers. 2 from glycolysis, bunches of electrons. I don't really care too much about the number of electrons, but notice bunches of electrons being gathered. 2 ATPs from glycolysis, that was substrate level. 2 from citric acid cycle. And again, the theoretical, 26 to 28 from this last process that I was talking about, depending on how many leak through. So overall, if you do it aerobically, you get 30 to 32 ATPs from the same glucose that if you do it anaerobically, you only get 2. All right, so we get the 2 plus, if you're anaerobic or doing lactate, you only get the 2. Make sense? One more quick slide, and then one more slide that might be just a little bit longer than quick, okay? All right, we get energy from all kinds of things. It's not just carbohydrates. You saw that picture in the middle. This is nothing more than kind of one of those FYI pictures, right? You get energy from oxidizing fatty acids and amino acids. And what I'm really trying to show here is kind of like this I-4 analogy that I gave you. The things that you eat enter at different spots. So if you look down below, what is all that in the red? That's the citric acid cycle, and that's different amino acids and where they enter. I don't need you to know that, but just know that there's more than one way to get energy from food. But basically, that's why that part in the blue with the circle was in the middle of that schematic. All right, thank you. Last slide. Everything has to be regulated, right? Everything. You can't be respiring all the time. ATP only sits around for a couple of minutes. Why make extra? So what's the best thing to regulate something? What's the best way to do is feedback. The best way to regulate the production of something is to stop it when you have too much of something. How do you know to stop pouring water into a glass? When it pours over the top. Okay, when you have too much. So when you have excess, remember feedback is excess, ATP, it shuts down one of these enzymes up here in glycolysis. This one is what, phosphofructokinase, right? Makes perfect sense. And when ATP starts running down, nothing inhibits the enzyme. Citrate can also be an inhibitor. Citrate is one of those things made in the citric acid cycle. It inhibits the same enzyme. Okay, that's right. And then finally, something we haven't talked about, this is what we call this whole idea of a feedback loop. When your energy level gets real low, you can sometimes break ADP into AMP. That's with adenosine monophosphate. That's real low. You generally don't want to get that low in energy stores. You never want to go lower than ADP to ATP. But if a lot of AMP begins to gather up in your body, that will actually stimulate the enzyme. You can inhibit an enzyme chemically. You can bind to it and cause it to have a much better fitting active site, and that would actually stimulate the enzyme, right? So you can cause things to speed up and slow down. But it's all based on most of it anyway on ATP, isn't it? All right, I'm done. Let me stop this.